My name is Denton Parcells, and the research I'm presenting is over sexual selection in response to varying levels of cultural eutrophication. Basically, the main question we looked to address with our research was how does human-induced eutrophication affect ecosystems? This has mainly been addressed in the past through community-wide studies and biodiversity in those ecosystems, but we were looking to address how does that affect specific species and traits in those species. And those specific traits that we looked at are sexual traits. Sexual dimorphism, specifically of sexual traits, is where the two sexes show different traits that are used for mating. These two traits come in two types. Those are either weapons like deer antlers, where the males physically fight each other, or ornaments like the plumage of a peacock, which is used to attract a female. These traits are rich in information since they are large and expensive to make. And since they're expensive to make, this gives females information on how successful that male is. If the male can make those large antlers or make extravagant peacock feathers, for example, the female can see that that male is good at getting resources in the environment, or they're resistant to parasites, for example. But either way, that male has desirable traits that should be passed on to their offspring. And since these traits are large and expensive to make, that makes them very sensitive to environmental stress. So another question we need to talk about is what is eutrophication? There are two types of eutrophication, either natural or cultural. Natural is what happens normally in an environment with runoff from the surrounding environment, and those resources go into nearby streams and lakes. Cultural is what we're looking at, which is runoff from like fertilizer and agriculture, or even lawns. When lawns get sprayed or have fertilizer put down, if the rain comes, that those nutrients can also be washed away into the gutter systems and into nearby streams and lakes. The picture on the right here shows an experiment from the 1970s where they put up this physical partition right here and added a whole bunch of carbon, nitrogen, and phosphorus into the lower section of this lake. As you can see that there are huge algal blooms where they added those nutrients, and the top side is still normal. And that shows just some of the impact that eutrophication can have on these ecosystems. This is also seen in the Gulf of Mexico with runoff there. Large algal blooms happen in that area that cause dead zones where fish don't get enough oxygen. And along with that, eutrophication is expected to disproportionately affect sexual traits due to the expensive nature that I talked about. And again, those consequences can have community-wide and ecosystem-wide impacts. Some of the hypotheses that we generated when coming into this study were that higher levels of eutrophication, mainly due to phosphorus, leads to larger sexual traits. Males and organisms are able to use the excess nutrients to help build larger sexual traits. We also hypothesized that higher levels of eutrophication will decrease sexual trait variation. The two pictures, the picture and the graph I have to the side, kind of depict that. This graph shows a study done on stock-eyed flies, which is shown in this picture right here. Males and females have sexual dimorphism, where the males grow these really long eye stalks that they wave around to attract females. And the females have much shorter stalks. And so in this study, family lineages were grown on different mediums, either corn, spinach, or just cotton wool, where corn has the highest nutrients and cotton wool has the least amount of nutrients. And you can see in the darker lines that when they are grown on the higher nutrient medium, all of the males are very densely packed together on stock size. And so when you go down to cotton wool, there's a much greater disparity between each of the lineages. So basically, when there is higher eutrophication, we expect to see the same trend as in the corn-grown stock-eyed flies, that all of them are very similar. And in the cotton wool, they're all very different. So when there are less nutrients, this allows the females to easily pick out which males are successful, which would be the ones at the top, and the ones at the bottom are not successful. But when there's that much eutrophication, females have a harder time selecting a mate since they all look so similar. And so we tested these hypotheses by comparing amphipod populations that live in lakes experiencing different levels of nutrient pollution. We chose amphipods for multiple reasons. Amphipods are found in most aquatic systems, so different populations are subject to many different environments. They're also important primary consumers, so a change in their population can cause an ecosystem-wide reaction, whether that's positive or negative, and that's called a trophic cascade. Amphipods also have sexual traits that are known to be sensitive to nutrient levels in their environment, specifically phosphorus. And so the sexual traits that we looked at was the second antenna here and the gnathopod, or their claw. This is a male gnathopod or amphipod, and their gnathopods are 15 times larger than in the females. Another reason we chose amphipods is their mating patterns. Amphipods have two main mating processes, either sexual conflict or female mate choice. In both of these cases, the success of the male depends on the trait size in the gnathopod and the second antenna. In sexual conflict, the males pair up with these females and actually direct their movements, and females try to resist a pairing duration too long, otherwise they receive negative repercussions. In female mate choice, the female also chooses to pair with males that have larger trait sizes. Pairing behavior also allows us to identify successful males in the ones that do have pairs, and unsuccessful males in the ones that are not paired. This allows us to, anytime, go out to the field and collect a sample of amphipods and see right then and there which traits are successful, how big the amphipods are, and in turn, how much nutrients are affecting their environment. The way we did this, both paired and unpaired individuals were collected along different transects. But this is only data from paired males currently. Different species of amphipods live at different depths, so this allows us to see how different species react to different pollution levels across those different depths. Amphipods were collected from lakes in Pennsylvania, and there were thousands of them that were collected. All of these lakes were formed from glacial retreats around 10,000 years ago. So they're very similar in the way they were created and how long they've been around. And the main difference between all of them is the nutrient levels and the agriculture in their area. So this graph to the right shows the lakes that we have done so far and the ones that are filled in, and the ones that we haven't done so far. And all of these eutrophication levels are measured in phosphorus totals. So LaBeouf being the highest and Sandy being the lowest eutrophication levels. Paired individuals' sexual traits were dissected, imaged, and measured using computer software, and we use graphs to analyze the results. So these are two graphs that address the first hypothesis I went over, that males collected from more nutrient-rich lakes would have larger sexual traits. The graph on the left shows the gnathopod size per each lake, and the lakes are listed from left to right in order of eutrophication levels. Same with antenna size over here, and in both graphs we do see a trend line going upwards just as we expected. These results are preliminary, however, and we have not collected all of the lakes or all of the amphipods from the lakes, nor have we run any statistics on these. We are just showing them as we have them. The second pair of graphs pertains to our second hypothesis that there would be more sexual trait variation in lakes with more nutrients. We measure this as allometric dispersion, which is a relationship between the trait size, which is gnathopoid and antenna, in relation to the body size overall in those individuals. As you can see in the graph, we don't have a correlation with any of this. There isn't a trend line that we can follow, but hopefully we'll be able to follow up with that with more data in the future. So in conclusion, amphipods are a big part of the food chain, and if they suffer, other organisms do as well. Changes in the fitness of amphipods can have detrimental effects on the rest of the ecosystem, and analyzing the source of this problem can help us prevent that in the future. The food chain right here kind of shows a depiction of how important amphipods are in their ecosystem and the amount of interactions they have with other life in their environment. Specifically, algae is a big one because amphipods help control the algae population. And too much algae can cause bad things. To go further with this, unpaired individuals need to be measured to provide a complete picture on exactly how nutrient pollution can affect the males and the sexual relationships they have and the trait sizes that they have. Again, those larger trait sizes allow males to make larger weapons, which can overcome female resistance to mating with greater ease and can in turn cause the decrease in population fitness that we don't want to see. That's all I have today. These are all the people that have helped with the project so far with either imaging or collecting or whatever it may be.